New York Bio brings you our CEO Roundtable on the rise of gene therapy in New York City from our 2021 annual meeting, sponsored by Fox Rothschild and featuring Rachel McMinn from Neurogene, Nolan Townsend from Lexio, Zandy Forbes from Mira Gene Therapies, and moderated by Michael Glynn from Fox Rothschild.
If we just look at Zolgensma, for example, it just amazing efficacy, and on a personal note is really what crystallized in my mind that I needed to be involved in gene therapy, and I founded Neurogene 2 months after reading that paper. So this really is a powerful technology. But I think over the last number of years, it's become clear that you can't just copy paste everything that happened with Zolgensma onto any gene with the same delivery mechanism, with the same manufacturing approach. I would say just to sort of start off, a lot of these programs, have started in academia, which has been a wonderful, wonderful thing, but the vectors were not designed with the end in mind of commercialization, and so you've had to see a lot of sort of backtracking, even in the middle of a clinical trial, to revamp a process. You're seeing the route of delivery not necessarily well thought through. Is systemic delivery for every single disease really most appropriate? We've done our own studies and shown no, that especially if you're treating CNS diseases, you need to be more thoughtful about how you deliver. And the manufacturing process itself is very complicated. There's a lot of things we don't know. And the list goes on. There's also endpoints and where do you intervene in disease. Not all diseases are going to be, just like with every novel technology. If you think back to monoclonal antibodies, you couldn't just take a monoclonal antibody and cure the world with every single disease with that one approach. So I think we have an incredibly powerful technology. We're as an industry, figuring out exactly how to use it safely, which patients, which delivery methods, how to clean it up, how to characterize it.
In a way that just about works. So as an industry, we're really building those tools, and from my perspective at Mira, it was essential for us to build every element of that toolkit, and that included manufacturing. And today, again, for AAV and Lenti as well, there's no clear-cut, well-known manufacturing process that you can just bring an AAV and manufacture it. So there are challenges to having a platform process that can rapidly allow you to have a process that can be scaled during clinical development. Because in gene therapy, it's a very complicated product, and ideally, from a regulatory perspective, you really want the product that you put into people at the IND stage to be produced in a very similar, if not identical way, to the commercial product. And as an industry, there are challenges there because of speed. It can take 5 years to develop a process and build your facilities, and yet you want to start your clinical trial in 6 months. There's a capacity constraint, but there's also a knowledge acquisition constraint for each of those individual companies to be able to get to the standards required by regulators globally, and in early stages of gene therapy clinical development to be of the standards required for commercialization. Because if you change your process or your facility halfway through or partway through, you run the risk that your product will not be considered by global agencies the same product. And that risk means that you may have to go to the very beginning of development again and start your IND. So I think there are challenges, but individual companies and the industry as a whole is starting to develop more and more tools to address some of the things that we've just mentioned.
I mean, I think if you look at this from a broader picture, that if you have 100 gene therapy companies that all establish their own manufacturing, you could imagine the inefficiency that will come from that over the long term, and then you could envision that significant amount of consolidation in the gene therapy sector that consolidates manufacturing onto common platforms and then creates the cost synergies that allows manufacturing costs to come down over time. So will we see that happen over the coming decade or decades? Probably. But I think in the near term, a lot of companies will continue to go the direction of what was described and kind of control their own processes. So I would almost separate sort of high-dose systemic gene therapy from a lot of the other work that's ongoing and from a patient, let's say, safety risk perspective. But even with high-dose gene therapy, I think we're looking at this risk-benefit trade-off for these very serious diseases like DMD and myotubular myopathy and others like that, where patients are making this kind of risk-benefit trade-off today. But I also think that's the place where novel vectors, novel promoters, some of the second-generation AAV technology could come in to help us address diseases like DMD and others with technology that is better suited to the disease. So I think there's sort of almost a line, and the FDA panel almost clarified this for everyone in a way of sort of 1E14 vector genomes per kilogram and above systemic is viewed to be, quote, high dose. And I think, below that, you don't see some of the safety issues that were described in that session. So I think that's what I'm defining as quote, high dose gene therapy. And I think that's where you've seen the majority of the...
That being said, I think, again, additional delivery routes where you don't have to give such huge amounts of exposure directly to the liver can avoid some of this liver overload. So if you can do that just by using an intra-CSF administration, again, if you're in the CNS space, it probably makes more sense than delivering such a high load. And there's also second generation technologies that can be built into the vector that we've been working on that really tamp down unwanted overexpression of the transgene. And I know that's being done in other labs as well. So I think it can be a multi-pronged approach.
And I think there's a risk benefit that, a lot of patients are considering, a lot of companies are considering when, entering into clinical studies at higher doses. And, it's really a race to deliver a treatment for patients with diseases where, their life expectancy will likely be cut short. But in the future, I do think there are, technologies coming that can continue to do this in increasingly safer ways.
Where in the eye, as you get to higher doses and you start seeing the higher likelihood of immune response, while that immune response may well be manageable and tends to be manageable, it obliterates activity. So we're still at a stage where we are trying to get the potency of our vectors right enough, so that means strong enough in a cell autonomous fashion to be most effective at the lowest possible capsid burden. So if you can control the gene expression that's coming off of your gene therapy product, it's not only useful for things that may be toxic or things that may be delivering drugs like peptides and hormones, but in gene therapy in general, it may be able to help you even the dose that you're giving to everyone so that you'll get a more consistent response when you're delivering at the current high, somewhat variable doses.
Those diseases, but I think it goes beyond that, even to diseases we're treating today. We could treat them in totality in a more cost-effective manner. So I use the example of hemophilia. So today, a hemophilia patient costs the healthcare system in the US about $400,000 a year for the entirety of their life. This is with factor replacement, extended half-life treatment, or Hemlibra, whichever treatment they have. So even if the gene therapy is $2 million one-time treatment, and that's, let's say, covering about 5 years of their existing treatment, year 6, year 7, out to the, presumably the useful life of the gene therapy, which we don't know, what that is yet. The healthcare system is saving on the treatment costs for those patients. So, you can replace even, existing treatments for certain genetic diseases today with a gene therapy treatment that will over time save the healthcare system significant amounts. But it actually does not stop even at genetic diseases. You can treat acquired diseases with gene therapy as well, with therapies, for example, vectorized antibodies. So this is delivering the genetic coding sequence to a monoclonal antibody into the cells in an organ so that the organ persistently expresses the antibody. So this technology can replace today's monoclonal antibodies with one-time AAV-based treatments. So this can replace a lot of treatments for even acquired diseases today with gene therapy-based approaches, single administration, functional cure to the disease.
Yeah.
Any thoughts
So
on
that was really nice setup for me, actually. Yes. As I mentioned, when we started Mira, we really started the company to optimize how you use gene therapy to not just address genetic diseases and use gene replacement, but to develop this new modality to cost effectively treat many large indications which weren't necessarily treated as well as they can be today. And one of the key technologies that we developed to enable us to do that is the gene regulation. So today at Mira, our preclinical pipeline consists of a multitude of vectorized antibodies, such as PCSK9, IL-17 antibodies, a whole range of those antibodies, which we can deliver the viral vector into the animal or the person, and then with a small molecule, we can switch that on to the same level that it would be on if it wasn't regulated, and we can replace an injectable antibody with a pill. So you have a one-time gene therapy treatment, and then you take a pill to activate the right amount of antibody that is going to address your particular disease. This is a really interesting new technology because it's a delivery device for otherwise hard to deliver biologics whose physiological function and therapeutic application is really well known. The gut peptides are one of them. There are many hormones and peptides all across endocrinology and metabolism and psychiatry that are really hard to deliver either locally or at the right time. And we can now do that by delivering the gene and switching them on with a small molecule.
I think, you know, there's so much promise, and my fellow panelists have talked about some of the places where gene therapy could be used. I think over the next couple of years, though, there's a very close watch by regulators, and that, you know, my personal view is that going into much larger sort of mainstream indications, we're not there yet. We really do need to better characterize the products. We need to better understand the safety profile. You know, we've seen a complete response letter, for example, even for hemophilia, which is a devastating disorder. There are approved treatments. And, you know, patients, I think from a regulatory perspective, the risk benefit wasn't there yet. One of the challenges is also just, you know, diagnosis and how early can you find these patients? And do we really have an infrastructure and even a bioethics system that supports early diagnosis? You know, I think the next couple years is going to be a little bit of, you know, putting in the hard work to really, you know, fully characterize. Luckily, you know, there's been nothing really particularly new that, you know, that was presented at this FDA panel meeting a few weeks ago. There was no, you know, smoking gun, you know, sort of big giant bullet that's going to take gene therapy down. I think we all kind of came together, saw where the FDA was headed. But, you know, they want more monitoring. They, you know, want us, right now we're limited by serially dosing patients. You dose patient 1, you wait 30 days, or depending on your trial, 45 days, and then you dose patient 2. That is slow, you know, as an industry, and it does make it a little bit challenging. So I think all of these visionary places for where gene therapy can go is, you know, I can see that vision too, but I do think there's a lot of hard work to get from here to there.
Thank you for joining us for our CEO Roundtable on the rise of gene therapies in New York City from our 2021 annual meeting. Thank you so much to our panelists and to Fox Rothschild for sponsoring the event. If you want to learn more about us, follow us on Twitter at NewYorkBIO, follow us on LinkedIn, or visit our website at www.newyorkbio.org.